welcome to Medical Dialogues Journal Club, your daily dose of health and medical news. I'm Mr. Zaman, and today I'm going to talk about genome reconstruction of centuries-old E. coli using fragments extracted from an Italian mummy. An international team led by researchers at McMaster University, working in collaboration with the University of Paris Cité, has identified and reconstructed the first ancient genome of E. coli using fragments extracted from the gallstone of a 16th century mummy. The discovery is published online in the journal Communications Biology. E. coli is a major public health concern causing significant death and morbidity, yet is not a source of pandemics. It is known as a commensal, a bacteria that resides within us and can act as an opportunistic pathogen, infecting its host during periods of stress, underlying disease, or immunodeficiency. Its full evolutionary history remains a mystery, including when it acquired novel genes and antibiotic resistance, say the researchers. Unlike well-documented pandemics such as the Black Death, which lingered for centuries and killed as many as 200 million people worldwide, there are no historical records of deaths caused by commensals such as E. coli, though the impact on human health and mortality was likely tremendous. Modern E. coli is commonly found in the intestines of healthy people and animals. While most forms are harmless, some strains are responsible for serious, sometimes fatal food poisoning outbreaks and bloodstream infections. The hardy and adaptable bacterium is recognized as especially resistant to treatment. Having the genome of a 400-year-old ancestor to the modern bacterium provides researchers a point of comparison for studying how it has evolved and adapted since that time. The mummified remains used for the new study come from a group of Italian nobles whose well-preserved bodies were recovered from the Abbey of St. Domenico Maggiore in Naples in 1983. For the study, the researchers conducted a detailed analysis of one of the individuals, Giovanni de Vallos, a Neapolitan noble from the Renaissance period. He was 48 years old when he died in 1586 and thought to have suffered from chronic inflammation of the gallbladder due to gallstones. The technological feat is particularly remarkable because E. coli is both complex and ubiquitous, living not only in the soil but also in our microbiomes. Researchers had to meticulously isolate fragments of the target bacterium, which had been degraded by environmental contamination from many sources. They used the recovered material to reconstruct the genome. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.